Hello! Have you ever considered that perhaps you could take your undergraduate project on an international stage? Well, this summer, a group of undergrad students had the opportunity to present an academic poster at the British Ecological Society Aquatic Group Conference. And in this video, we are going to show you how this came to be.
This poster was based on data collected by 15 undergrad students doing their final year project at Silwood Park. And within the team-based project, they were divided into 4 different teams that were looking at different levels of biological organization, ranging from individuals all the way to ecosystems.
These projects were supervised by Professor Guy Woodward and took place at Silwood Park, using the site's large-scale mesocosm system with 96 outdoor artificial ponds, those with different levels of warming and chemical combination.
So we were more focused on the individual element, so the impact of warming on 2 focal species. We had Acellus aquaticus and mayflies, or Chlorion dipterum. And so essentially what we did is we had a series of mobility trials where we essentially noted where an organism was every 5 minutes on a Petri dish. And then at the end, we had a bunch of lines, and connecting those lines, you could tell the distance that an organism traveled. And using that information, we could see the impact of temperature on the distance that these organisms traveled.
We focused on interactions between predator and prey, and this consisted in running trials where we would use aquaria and we would put one predator with prey in it and then leave it for 24 hours and just count how many prey had been eaten, and then we test that at different temperatures. So we were testing temperature effects on the amount of prey eaten.
So our team had 4 students, but we were divided into 2 pairs. So one of the pairs focused on the benthic organisms, the macroinvertebrates and the mesocosms, and me and my lab partner focused on the pelagic zone. So we focused on the zooplankton present in the mesocosms, and we focused on community level changes due to warming and chemical pollution. So we essentially looked at changes in body size and community composition of these different organisms.
Our team want to investigate how do warming and chemical legacy effects affect the freshwater ecosystem. So we were divided into 2 groups as well. 2 of us study the green pathway and the other 2 study the brown pathway. And for the green pathway, we measured the change of the primary productivity of the primary producers. And I think for the brown pathway, they measured the decomposition rate and also the CO ratio.
So as a GTA, I help to coordinate the projects all the way from planning to troubleshooting experiments and being also the day-to-day supervisor of the students. And I must say, being in the process of going all the way from the beginning and creating the projects to seeing the students collect all of the impressive amount of data and then develop it into their final piece of writing was really, really incredible experience to be part of.
Although I wasn't actually part of the team-based projects themselves, I joined Professor Woodward as a Europe student over the summer, and this allowed me to get very up and personal with the mesocosms, keep the data collection going from the previous team-based project students, and help develop some resources for the next year. And this is actually how the whole poster idea came about.
Alongside my final year project, I also worked with Andra over the summer on Europe. Professor Woodward told us about an opportunity to present all the team's findings at the BES Aquatic Group Conference and encouraged us to apply. Even though the deadline was tight, we jumped straight to it. I helped write and submit the abstract, making sure that our research story was clear, engaging, and accessible to a broad audience.
And this is when the real work began. To put the poster together, we needed to make sure we're familiar with everyone's thesis. So Andra and I went through all the final year thesis, pulling out the key results and visualizing how different organizational levels can link together. We needed to make sure we didn't overload the poster with too much information, so this required deciding what was the most relevant and representative findings.
So when it came to the poster itself, we made sure that we applied everything that we had learned in our undergraduate degree, and we made sure that it was visually pleasing, easy to follow, and that it had all of the information that it required for the whole story to make sense.
When we were happy with the poster, we sent it back to all the 15 students to check and confirm their co-authorship. And all that was left now was sending it off to the conference.
The conference itself was amazing. We were greeted with tea and coffee in the morning to wake up, and then followed a day full of talks and interactive creative studies, which allowed us to learn more about the research that's going on in the aquatic world, and it gave us an opportunity to talk to academics which were actively researching in the fields that we were interested in.
We heard from multiple scientists who gave talks on their research on topics such as blue carbon or aquatic microbial communities, to even how we can turn science into art, which was a great experience. And I even had the opportunity to give a lightning talk on my undergrad project, which was on the impacts of pesticides in UK rivers. So overall, it was an amazing experience, and I'm really happy that I had the opportunity to join.
We would like to say a huge thank you to Professor Guy Woodward for supporting us with the project and the conferences, and to the British Ecological Society and Linnean Society for hosting these interesting and stimulating conferences.